Hello everyone, my name is Angel Fuentes and today I'll be presenting on Adapt to Brain Health for MCDBIO50 and I hope you enjoy my presentation on neural stem cells. So let's get right into it. And so let's imagine a hypothetical scenario. Let's imagine that you are a luchador, you are in the pinnacle of your career. You are one of the greatest wrestlers known to man in the WWE or the World Wrestling Federation, and then you're at the climax of fighting the Undertaker when the Undertaker, boom, uses his tombstone driver and hits you right in the head with a severe concussion. And so, what is next in your path of recovery? What happens now? And so, the doctor then gives you the diagnosis. You're given a traumatic brain injury, which is defined as an injury to the brain, usually caused by some sort of blunt force trauma or by a collision internally or externally that causes the brain tissue to begin jolting around within the skull or cranium and compromises brain tissue and could cause permanent neurological damage. And so there are three main hierarchies when it comes to traumatic brain injuries, and those are mild concussion TBIs, which are the most common and can produce brief alternations between consciousness and being in an almost dazed state for maximum 30 minutes, and a state of confusion that may remain for the entire day. And so these are usually caused by sports injuries like football or baseball. So these are more common than the ones that are less severe. But the ones that we'll be mainly talking about are moderate traumatic brain injuries and severe TBIs. And a moderate TBI is similar to a mild one, but instead the state of dazed consciousness lasts for more than 30 minutes, and the overall confusion can last to upwards of a week. And for a severe TBI, this can be even worse, in which the individual loses complete consciousness from a knockout, and the injuries are typically associated with changes to the head and brain with additional neurological compromises and personality changes. So that's definitely the one we'll be looking at, and the type of recovery that we can see using neural stem cells. And so, even though there are these type of hierarchies, there are two main categories that both of them can be defined through, which are uncomplicated TBIs, in which even if there is a severe or moderate TBI, when undergoing a CT or MRI scan, there's ultimately more mild or regular brain activity still being undergone, in which there are no significant physical changes like internal bleeding. But then there are complicated traumatic brain injuries in which there could possibly be potential hemorrhages or neurological compromises seen through those CTs or MRI scans. And there's, of course, closed TBIs in which the internal structure of the skull is not compromised or not fractured, meaning that there are no blunt force traumas that could have penetrated into the brain, and open traumatic brain injuries in which a blunt force trauma could have penetrated through the skull and into the brain. So, and that's something to definitely consider when talking about these traumatic brain injuries. And so, now that you're given the diagnosis, what can you really do? And so, for the majority of mild traumatic brain injuries, fortunately, there are means of recovery, and that is rest. And that mainly includes all the other types of TBIs as well. But when it comes to moderate severe TBIs, there could also be the potential for blood clot removals due to the fact that the blood that accumulates within the skull could then coagulate and turn into blood clots. It could further compromise brain tissue that was already injured. A skull fracture repair, in which, again, for an open traumatic brain injury, the pieces or fragments of the skull are then placed back into the brain or placed back into the head in the correct formation in order to make sure that no additional damage is done, and also intracranial pressure reduction in order to make sure that if that blood begins to accumulate, or if there's like cerebrospinal fluid that begins to accumulate within the injury, that the reduction drains all of that fluid out to make sure that no further damage is done. And as you can see, there really isn't any cure or ability to truly recover from this injury other than through rest, because otherwise you would take anticoagulants, muscle relaxants, and antidepressants. But there is a new treatment being discussed lately, which are neural stem cells. And neural stem cells can differentiate into neurons, glial cells, and oligodendrocytes in order to differentiate within the brain and occur naturally within the dentate gyrus of the hippocampus and the striatum, and are can be extremely useful in order to develop and differentiate into those neurons that they can reroute neurological impulses or electrical impulses from the compromised brain tissue into different parts of the brain so that the brain could recover not by full regeneration but by rerouting certain neural impulses. And so there are four main steps when it comes to a neural stem cells differentiation, and that is proliferation, where it goes through the usual phases of mitotic cell division as it's still an undifferentiated stem cell, and then it goes into migration, in which neural stem cell then becomes somewhat defined as a progenitor stem cell and is able to move to its correct location as a neuron uh, according to specific uh, physiological factors or environmental factors around the neuron and can move at around 100 micrometers per day until it reaches its final destination according to, once again, those chemical factors. And there's finally differentiation, in which the cell finds its destination, and this involves axon growth and dendritic arbitrization, with an additional absorption of means by which the neuron can communicate with other ones. And then finally, synaptogenesis, in which the neuron, and this is the most significant aspect of neural stem cell differentiation, in which the neuron then attaches to other neurons around its area, and this occurs through the neuron's axons and dendrites with growth cones that guide the cell to the target location and specialize into a specific neural function according to the other neurons within the neural stem cells location. And these can be utilized either intravenously or even directly injected into the individual's brain, and have been tested on mice and have shown that at maximum in 1 week after the injury can have a 9 to 25% success rate and have such neural stem cells be retained for around 5 months. And although this can seem pretty low numbers for right now, it is pretty significant for the progression of this treatment later on in the future. And so even though this is a very innovative and incredible technique, there are some side effects. There is the fact that immunosuppressant side effects could decrease the overall immunity of the person, even though those immunosuppressants prevent a bodily rejection of those stem cells. Sometimes those neurons can move to the inappropriate parts of the brain that could cause rerouting in the inappropriate areas that could cause mental diseases like schizophrenia. And those undifferentiated stem cells could continue to divide or migrate to the gray matter of the brain and cause other neurological complications as well. And of course, as I was talking about with the first bullet point, there could still be the possibility of bodily rejection and severe symptoms and medical situations that could stem from that. So even though this is a very progressive treatment, there are many consequences from this as well. However, there is the possibility of electrical stimulation for these neural stem cells that can both help the neural stem cells differentiate into the correct neurons for that injury and also go to the right place of migration in order to attach into the correct place in the brain. And so, once again, electrical stimulation of NACs and localized brain injuries are stimulated through electrodes or through a transconductor enzyme called PEDOT, which is both biocompatible and able to conduct that electricity to those neurons as needed. And during this research that I discovered, they used cross-linked PDOT films and used a cell culture of neural stem cells with certain growth factors for, once again, immunosuppressants, but also growth factors for the correct differentiation of those neural stem cells and other factors to make sure those stem cells maintain their ability to differentiate throughout the experiment. And so the cell culture was implemented into an environment similar to cerebrospinal fluid, which I thought was interesting, and was given, of course, the famous fluorescent protein in order to see if those proteins or cells or certain proteins of those differentiated neurons were being expressed as the experiment went on, and PCRs in order to see if successful differentiation migration occurred. And they used a, I believe, 1-volt alternate conductor in order to see if implementing electrical stimulation would help with differentiation. If you see right here, there are successful results in which an increase in the amount of voltage caused the frequency of the fluorescent protein to be exhibited, meaning that differentiation occurred. So pretty awesome stuff. And so again, the results were that it was successful. However, there are certain issues when it came to the experiment, both ethical and when it comes to its innovation. And it's that if those electrical stimulations exceed 1.2 volts, then electrolysis could occur, as shown in some parts of the experiment. And one thing to note is that in the experiment, they already used a 1 volt voltage, so going above even slightly could compromises in the cerebrospinal fluid that could lead to further injuries. And there's also the fact that I think from 7 to 9% of these surgeries could lead to a neurosurgical hemorrhage, which once again could lead to even worse complications to this type of treatment and injury. But overall, I mean, this is still like an incredible form of treatment that's being used, and it becomes really innovative in order to help out what seem to be like impossible injuries, right? When we think about the brain, we think about something that's permanent, but this shows that there could be future paths. So I really thank you for listening to this presentation and having a zap to your own brain of this information of electrical stimulation to neural stem cells. Thank you so much.